That's a great question. In human, I don't think that's been looked at quite well, mostly because postpartum tissue and processing is technically difficult, but also getting the sample itself is also challenging. And then in terms of where it is, I think there's very strong converging evidence showing that it happens in the prefrontal cortex where we study. It most definitely also happens in hippocampus. There has been suggestion that, maybe not everywhere loses synapse. In other places, there could be compensatory changes. A couple of studies have found that, for example, in the amygdala, maybe you actually have an aberrant, too much synapses
Hmm.
there. So I don't think, everywhere you're losing the synapses.
Yeah.
Maybe one better characterized is just the circuit connectivity has been altered. But in the prefrontal cortex that we looked at, you tend to have a loss of synapses.
Hmm. So there's different changes in a bunch of different parts of the brain. It's not like everything is happening in one direction in one place, but the prefrontal cortex in general is often heavily implicated as a place where quite a bit of change seems to happen in response to things like chronic stress. And when you look at the cellular level in animals in general, in the prefrontal cortex, there seems to be a loss of spines, a loss of excitatory input onto at least certain cells.
That's correct. Yeah. So, I mean, this has been shown in different ways. You can, take tissue and look at them and fix tissue. In my lab, we specialize in live animal imaging. You can also look at synaptic protein. In some of the other recent study, people have also do sequencing of transcript. These are also can be done from human tissue, where they also can do large scale sequencing to find synaptic changes. So I think all in all, really, there's a fairly good amount of converging evidence showing synaptic deficit associated with depression.
And so, okay, so when we see these structural and functional changes in animals that correlate with stress and depression like behavioral changes, how do things like typical antidepressant drugs affect these changes? Do they reverse them partially or completely? Do they lead to other changes? What do we know about the effects of, say, SSRIs?
Yeah, so it's commonly known thought that the SSRI and drugs that can be used for treating depression can lead to some restoration of some of these synapses. Uh, Now, it's funny you ask SSRI because I think SSRI is actually one that is not as well known. Arguably, there actually been more done on ketamine and psilocybin by
Hmm.
recent work. But the idea, and there has been several studies showing that the SSRI does increase synapses, but maybe not as strong as one would like in terms of the evidence.
Hmm. And what about sort of the dynamics here? One of the things
Mm.
SSRIs I think are pretty famous for is that, in a subset of people, they often work to some extent to relieve symptoms of depression. But very often, I think almost in all cases, when that happens, there's a lag. So it takes some amount of time. Is there any sense that the SSRIs are sort of working? What are the timescales of action here? Does it take some time for us to see changes in the brain after you start giving an SSRI to an animal?
That might be the reason why it's been harder, in fact, to see changes in the morphology of neurons in these dendritic spines after SSRI. I think what you point out could be a key reason why it's less often seen. Because in human, you're right, like SSRI would take 4 to 6 weeks, typically to start to become effective in a subset of people. And in animal, you would think that you also need chronic administration. It's not that 1 or 2 doses would lead to very dramatic changes. So it makes it very difficult to find these effects because you have to chronically administer it.
Yeah, it just
Uh,
makes it infeasible to do some of the measurements because it takes more time. You'd have to do more looking and more searching over a longer period, and all of the tricks that you use to record these things can be time sensitive and complicated.
yeah, and also I think the other thing to be aware of is in animal models, I think unlike in the human conditions, when you stress the animal would tend to exhibit the depressive symptom. But once you stop the stress, the animal will then tend to recover. So you also have some time course issue where you will stress the animal, then maybe you give it SSRI. If the animal gets better, is it because you gave it SSRI or is it because the stress is now gone? Maybe, yeah, all of these I think makes it perhaps harder to disentangle the differences. The other thing that we can get into a bit later is I think there's also a case to be said that maybe not all depression treatments are equal as well, and they don't necessarily need to rely on the same mechanism. I do think that there may be more similarity between certain types of treatment that are a bit more rapid acting.
Mhm.
For example, ketamine, maybe some psychedelics, or even uh, transcranial magnetic stimulation, versus other things that's a bit slower, like SSRI.
Yeah.